Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. In our last video, we have learned how to predict the ligand binding sites in a protein. In that particular video, we have used DOG site scorer and clips. I have mentioned there that they are predicting based on the geometric shapes and the grid information. Today we will learn how to use the embeddings or parameters of AlphaFold version 2. It is also called AlphaFold 2, and to detect the binding site information for a protein for a ligand. The program we will use today is called AF2BIND, or it's abbreviated for AlphaFold 2 bind. The authors of this program take the advantage from the contact representation of AlphaFold 2 and train a method which is called AlphaFold 2 bind. So far, the author have claimed in their research paper, or which is available, I will show you later, they have claimed that this program have so far outperformed all the available other programs which are working in the field. And technically this video is a kind of part 10 of the series computer aided drug designing. So let's begin. Okay, so this is the research paper which is submitted on October 18, 2023. It means that it's not a very old program, it's something new. The research paper I'm showing you is from the preprint BioRx 4 or BioRxV, whatever you called it, from the Cold Spring Harbor Laboratory, and preprints are here. Preprints means that this research paper will be in a peer review for some journal, but so far it is freely available here in the preprint. So if you have used AlphaFold 2 in your research, and until that time if you did not see any paper, this paper in any journal, so that's okay. I have shared you this preprint version. You can share or you can cite this preprint version if you want to read it. So you can click here for the full text, and the full text paper can be found here. You can see this is the full text paper. So okay, let's come to the point. So the program we will use is AlphaFold 2 bind, and if you want to use some classical programs like DOG and clips, so I have already covered a video in part 9, also in part 8, but in part 9 we have covered the comparison between DOG site scorer and clips server or clips database. So remember this AlphaFold bind is a kind of machine learning model these authors have trained it, and you will use it or we will use it. So also remember, currently we have AlphaFold 3 release last month. So as far as we don't have another update like AF2BIND, so when it comes I will share a video on AF2BIND. Supposedly it will be AF2BIND or maybe AF2BIND version, some another version, but so far we will use it on the AF2 or the AlphaFold 2. Okay, so let's start. First of all, we have to open the notebook I will share this link in the description section of this video. Now come here, create project. Now you will see binding site detection with AlphaFold 2 part 10. Click here. Now connect. So here is a brief introduction of this channel and about me. Okay, it's a simple and faster program. You can use it very easily. Okay, once it is connected, then you click here, install AlphaFold 2. Now it will install AlphaFold 2 embeddings, and also it will install the AF2BIND. Okay, so until it is installing, we are coming here that in how many ways you can run this program. First of all, you can run this program, use a PDB ID directly into this notebook. So first of all, we will use a PDB ID directly. So I will use the PDB ID which we ran in our last tutorial, part 9, that was 3W32. Okay, nothing else, just write a PDB ID, and for that video, for that structure, I know the chained information are A, but you can check it from the PDB whether it is A or B, so that is up to you. Okay, so just click this play button, but so far it is running, so that's why it will not start running. Let me pause. Okay, it's finished. Now remember, I will perform all the 3 steps, that how will you interpret your results, but most importantly, you can use this program in 3 different modes. First of all, you can directly provide the PDB ID like I did. Second, you can also use this program from Unipro accession number. What is Unipro and what are accession numbers, we will discuss it later. And you can also upload your manual structure if you have modeled it somewhere. Okay, it's done. Now you can see some information of 15 amino acids. Now if you want to see more, just click here, show, let's say 25. Now you are seeing the 25 information. If you want to use some filter, just click here. Total are 317 entries in this protein, so you can filter it. Okay, you click it, it's very easy. You will handle it. You can see from where to where, which chain, from which residue, etc, etc. But first understand this column is a rank column, this column is a chain column, which is A. This is the residue index number, this is the residue name, like in 1-letter code, like aspartic acid, valine, lysine, leucine, threonine, etc, etc. This is P-bind. P-bind, it's simply a probability bind. So 0.977 means 97% or 97.7% or roughly 98%. So it's a very higher score, 0.97. So it means you can decide this is one residue, this is the true positive value. Okay, coming down, the score will be substantially decreases. Okay, Now if you want to display the structure, so the color will be by confidence. What confidence? These are the colors, and the dark blue, it means one are near to one confidence, or if light blue is almost 75%, and the same way you can see if it's red it's 0%, if it's kind of whitish it's in the range of 50%, I would say. Okay, now if you see here, you can also see the table information here. For example, you want to see this residue, keep your cursor here. This is aspergine 842 and the score is 0.894. it means almost 89% or 89.4% is the p-bind value. It should be more because the color is more darker or dark blue. So yes, it is 855 SP, 97%. It should be a bit on the smaller side. Yes, 0.517, it means almost 51%. Okay, now I will not suggest you to use this structure in your papers or thesis, because if you use any program to cut it, actually you are losing the DPI. And the journals I have covered in some other video that the minimum is 300 DPI. But when you use some sniffing tool and to cut and paste, that way you are taking, I think, 96 DPI roundabout. So 96 or 100 DPI is not a good picture. So the question is, what will you do? You have already these information of these residue. Now you can save this table from here, or you can take a screenshot from this table, then use some program, Discovery Studio, Chimera, Pymol, VMD, whatever you are using. I'm not stressing for a particular program. So whatever you are using, use that program and make a beautiful figure according to your paper or whatever you want. Okay, you can also download the predictions here, just run this cell. Okay, that's it. So this was the analysis if your receptor protein is present in the PDB. Now coming here, use Unipro accession number into this notebook. Okay, what it means, let's say this is a Unipro. What is Unipro or universal protein, whatever you can say? So remember, Unipro is a database of protein where you can find information about protein, amino acid sequences, some PTMs or post-translational modification. The structure is available in PDB or not, whatever, it's okay, and some other related information. So let's say I'm searching anything EGFR. So let's say, let me search this protein. Okay, because this is a bit smaller, so it will take less. So let's say this is my protein of 421 amino acid. This is EGFR adapter protein from Drosophila melanogaster. And when you click here, so click here the structure option. When you click the structure option, if you see any PDB stuff, it means the structure has a PDB or X-ray crystallography or electron microscopy results, but we did not see anything. You can see the structure database did not say anything about the PDB. So it means only one predicted structure is available, but that was predicted by the AlphaFold in automated run. Okay, now So let's say this is your protein, and you want to find out the ligand binding residues. So in that case, and we do not have a PDB for it, so what will you do? Just note down this accession number Q69, Q6NRO, not O09. In fact, you can copy it from here. Copy and just paste it here. Now run it again. So let me it will take 1 or 2 minutes, so until that I'm going to pause this video. Okay, welcome back. It is finished and you can see the result, but don't be surprised if you are seeing the p-bind value is on the lower side, because this is a different protein, that was a different protein. So I took it from the Unipro, just a random protein, just to show you how you will perform this kind of thing on using the how to say the Unipro accession number. Okay, and let me display it. Okay, now if you see, you will not see any blue thing, because the p-bind value is below 50%, so it is towards the white thing, like even it's something here. So remember, if it's below the 50%, you will not see a kind of those stick representation. Okay, one thing I want to address here for all of you, of course this structure you are seeing is predicted by the AlphaFold, but that doesn't mean that AlphaFold will fold correctly everything. No, this is wrong. AlphaFold cannot fold anything correctly. You can see the structure. If you ask me which part is right, so visually I can answer you that only this part, the middle part, this one, it seems correct. I'm not saying this is correct, but it seems correct visually. And these parts, you can see these wires, they are not seems correct, because, and what we call it disorder region. So I'm not going to make this video more extended and to bring it towards the to calculate the disorder region of proteins. There are too many servers available. So if you are seeing this kind of stuff for your protein, it means your protein has a disorder region. Now what will you do? Because if the program is guessing this part some value for ligand binding, so of course this is a disorder region. This is a disorder region. How you can say that this will be ligand binding pocket? Okay, so what will you do in that case? This is a question. If I am your supervisor, or if I suggest you, then I would ask you that first of all do some literature study, that which domain is this one. Normally a protein has many domains, so which domain is this one? For example, it's a catalytic domain. For example, it is a catalytic domain and it's present on the C terminus, whatever. Okay, now I will read some paper and I will find out that whether the ligands are binding to the catalytic domain or somewhere else. If the ligands are binding here, then as a supervisor I will ask you that okay, the sequence from here to here, I would say here, take this sequence, first make a protein model, remove this disorder region, make a good protein model with good confidence. Now calculate the ligand binding residues using AF2BIND or any other program. But how will you do that? Okay, so the answer is, in that case you have to upload your own PDB locally. Now the question is how you will upload your PDB locally. That is simple, just click here, remove this, also remove this. Now you have nothing. Okay, let me clear it, also clear this thing. Okay, you have nothing. Okay, now play this. Once you play this, you can see choose file, click, and let's say this is your protein. I name it this is some protein. Okay, I will relax 1 and I will look it. I think its chain will be A. Now once it is ready, so it will be I have uploaded a protein from my own computer. So let's say what I'm getting. I'm again going to pause this session for a while. Okay, so these are the results of the protein I have uploaded from my local computer. And now click here. Okay, you can see this is some different protein. So I had in my computer, I had used it for relaxation video which is uploaded, how to make a protein relax. So okay, now these are the top ligand binding residue, these three. This one is 60 ILE and this one is 42 leucine and this one is 39 leucine. Okay, now another thing you need to know, if you see the residue numbers are 39, 43, 42, so try not to use these residue number in your manuscript because these are not the real residue number. Suppose this was your protein which we have seen a little earlier here. So that protein, more than half of that protein was disorder. So what you did, you removed the disorder region. So the residue, let's say the total protein is 500 amino acid, you remove half of them. It means you remove the starting 250 amino acid. So now the model structure or the model protein, the first amino acid which you are saying is 1, it will not be 1. Technically it will be 251 because the 250 amino acids are removed. So in that case, when you write your manuscript, you need to mention that amino acid as a 251, not 1. But why this program says 39 or 42? Because the program is reading the model you offered or you gave. You can also make these changes in a 1 minute using Chimera. That's very easy in Chimera. Just search it there. It's easy. So you can change the residue numbers of your model before subjecting into the AlphaFold to bind, or the other way you can change this table. Let's say if this is 39, so 39 plus 250 which are removed, so this amino acid will become 289. Okay, so I hope I have cleared each and everything for you in this video, and you all will be able to calculate ligand binding residues before docking and using AF2BIND or using DOG site scorer or clips, whatever you want. That is your option or your choice. Or in this notebook you can use it in 3 ways. So I hope you will like and you will learn something new from this video. If you have not subscribed this channel yet, please subscribe, or if you have your friends, you can share this channel in that circle. It will also help them. And so far so good. Stay happy. Good luck in your research. Bye bye.